and they were absolutely horrible to look at because they were just miles and miles of wire and circuits, which you engineers might like, but I like my cardiovascular system to be more cuddly. But they were actually very brilliant, and from those he would put in known information and run them through his various loops and predict the outcome, and then do the experiment in the animal and show the outcome. That's what happened. It wasn't desperately fashionable on the British side of the physiology, but he has probably Guyton's textbooks are the most famous textbooks in the world. Or no, Gannon was, but Guyton, for cardiovascular physiology, I still use Guyton. And but it's sort of, like anything, you have big rows. I mean, Paul Corner, who was one of the most famous cardiovascular physiologists, just said hypertension was in the brain. It was all neurogenic. But he used engineering principles, but, feedback loops and controls. Mm-hmm. So it's been around in cardiovascular physiology for a very, very long time.
So basically, it's just on the realm of modeling, not in the realm of explaining physiological phenomena.
Well, it helped explain them. For example, it showed that if you took the pre-existing knowledge at the time, and remember that was pretty limited knowledge, and did a particular experiment, you would generate an animal that had high blood pressure. So I think it did do a lot of explaining. It showed that kidneys were important. It showed that salt was very important. He proposed that salt angiotensin, salt blood pressure, sodium output and blood pressure were directly related on a sigmoid loop. Anyway, that sort of thing. So it was very, very mechanistic and lovely.
No, I'm thinking in the realm, for example, just out of arbitrary example here, like requisite variety helped to explain why particular physiological activity is that way. So that sort of explainability I was thinking. I mean, is there that sort of practical application?
Well, I mean, trouble with physiology, and this is the trouble I'm having with chapter 4, is to really understand what's happening, you have to go right down to the signaling pathways in cells now. And then you have to build up. And of course, as you do that, you get, well, the article that I'm reading at the moment has got 1,248 references, and it only deals with renin-angiotensin system and cardiovascular physiology or cardiovascular renal. It doesn't go to particularly into brain and whatever. And I can tell you 1,200, it's just horrific. And one of the things points the authors make is that when you look at angiotensin signaling pathways, they have individual consequences depending on the tissue in which they're in. Mm. And this is a fairly new concept, that basically if angiotensin does this in the kidney, this will happen. If angiotensin does this in the kidney, it does that in the heart, even though it may use the same signaling pathway, presumably because of epigenetic modeling of the cells as they're formed. That's the only way I can do it. And they talk about it as becoming, convergent, divergent, and very diverse, which really goes well for trying to fit it into an argument about systems. I think that's
the term of very diverse. That would be maybe analogous to the variety that we are. For example, if you draw this analogy between, like, I'm just using very diverse for now, but and variety, and see whether we can draw some conclusions, or because of variety of X cause this variety of outcomes, and this outcome require this variety of things, and then now we can understand more why
Yeah.
kidney behaving this way or so on.
That's what
sort of
I'm
level.
trying. That's what I'm trying to do, is bring it down to, you know, the core signaling pathways, and then to key organs, and then to the whole animal. But I'm honestly going terrible slow on it, which I guess is not
I'm
surprising.
having the same problem in terms of getting across the relevant literature, which is quite enormous now.
Yeah,
But the other thing about modelling is that biology is pretty good at doing it itself as well. And if you look at the lung and you look at the cardiovascular system, the rest of it, it really has a capacity to remodel itself. And just so many chronic disorders that are associated with remodeling it has gone astray. And
I think you're right. I mean, you look at angiotensin in the heart, and if you block one angiotensin-associated protein, you don't get fibrosis, but you still get hypertrophy or something like that. I mean, quite frankly, my brain's aging and keeping it all in. But you get all these specific remodeling happening, and of course, that is key to a healthy body, is to remodel or adjust to a situation. And then from that situation, you create another set of problems that you have to deal with and model. I mean, you know, they say the gut has 4 trillion 4 kilograms of DNA, bacterial DNA, you know.
Wow.
the gut's got about 40 to 50 trillion bacteria.
Yeah, 4 kilograms of DNA, because I always think I'd be 4 kilograms thinner.
Yes.
We're actually quite insignificant in the game, really.
But when you start to think about that, you start to wonder how the hell you can actually single anything out as being as significant in understanding the human organism.
I think the total viral load that a human carries is probably 10 to 100 times the bacterial load. So there are about 40 trillion bacteria. So you can multiply that by about 10 and you get an idea of the viral load we carry. All of it effectively is quite innocuous.
But what I like is that 0.04% of the total biomass is virus.
Yes.
And we are only 0.01% of the total biomass.
Yeah, we have a quite distorted view of where we sit in the scheme of things.
So this planet is not a human planet, it's a viral planet.
Yes.
Beg your pardon?
This planet is not a human planet, it's a viral planet. It's literally the viruses have created us just so they keep themselves alive.
Well, of course, we've used the viruses to make us what we are.
Or maybe the way around, like
it's both.
They just created humans so that they can survive better for more variety.
It's all part of the glorious complexity of this planet.
The viral RNA is responsible for forming our placenta. I mean, viral genes that have been incorporated into the human genome form the human placenta. It's a gene called syncytin. And in fact, there's been an argument about it with COVID, and is it's very similar to the ACE2 receptor, which it isn't. So it's a silly argument.
But we've been pretty good at borrowing stuff. I mean, all of our mitochondria are effectively ancient bacteria that we engulfed because they're very good at handling energy. And so we're a composite of all of this stuff. But I think a really important issue, because I just picked it up then, is the distinction between viruses and many other pathogens. Viruses are fundamentally innate, are inanimate. And so I'm leaning more to looking at viruses through the eyes of chemistry than you would look at bacteria, for example, through the eyes of biology.
That's right. You know, people talk about these wicked viruses, but they're just a bag of chemicals, and they don't have a brain. Don't have a
So are
brain.
we, Darren.
Yeah.
We're just
It's funny,
egg.
but because when they bond in a living being, they behave differently, don't they? So they are that chemical, like Dettol or something, nothing. But then when it attaches to a human living being, then it starts to act like it's living. So this is the
Well-
emergence.
Yeah.
I think viruses adhere powerfully to the basic laws of thermodynamics, and just like any chemistry does. Bacteria, because they have to respire, have more characteristics of living than straight chemistry. I mean, they've got to carry around their own ability to produce energy, and they do that through iron as opposed to ourselves through bacteria, through mitochondria. And Gary, I sent you some PowerPoints, only a few of them,
Yeah.
a few minutes ago, which starts to look at some of it goes to the fundamentals and says, is there a thermodynamic driver that sits behind this virus? And I suspect there is.
Okay.
So,
Right. Okay. So Jenny's joined us now. So I think what we'd like to do first is we'd like to give you a brief on where we're going with the papers in terms of how we intend to publish. Jenny,
Yeah.
do you want to say a few words, and then maybe I'll go through the papers and say where we believe they're going to go.
That sounds good. Oh, thanks everyone. And sorry, I've missed quite a few of the last meetings. It just seems to have been in my workplace. We've had significant restructure and a significant increase in my teaching load, all of which is fine, but I didn't understand that where all the teaching was happening, and it seemed that it came at the last minute and tended to be on a Thursday afternoon. So then I became triple booked, and I apologise about that, but I think we're getting on top of it. Anyway, so I haven't met all of you either, but I'm a clinical pharmacologist in Newcastle, and I'm also a deputy editor on a journal called Pharmacology Research and Perspectives. And the editorial board met, I think, some time back and requested some themed issues for the journal. It's only a journal that's been around for about 2 years, and they've got an impact factor of 2, but really the journal wants to become more relevant, I think, and improve not just its impact factor, but its impact. It's a journal jointly owned by ASPET, so that's the American Clinical Pharmacology Group, and also the BPS, the British Pharmacology Society, and Wiley. So it's a 3-way journal. Anyway, I said that I was working with Richard and David Fedson at that time to look at COVID and treating the host, and we felt that probably a suite of papers looking at COVID and the therapeutic opportunities, or just general opportunities, I guess, but therapeutics also that we'd missed during COVID, were because we looked at this in kind of a siloed way and hadn't really had a lot of consultation and collaboration, and perhaps we could set a suite of papers up documenting that and how we actually should have looked at the biology and pharmacology and the social aspects, and how we should have integrated those. And I guess we started with a bit of a, probably in retrospect, actually in reality, because I looked at it yesterday, but a scientific plan around how we should have approached this from a scientific perspective. And we got a bit stuck because there was a lot of system stuff that needed to come into that. And Eugenie kind of introduced us to Felix and to Gary and the team to try and get some sort of systems approach on where we're up to. And then we realized that actually we need a lot more help than just the science and clinical input that we'd had. So that's why our group and the plan became bigger. And that's looking all pretty cool, actually. But the problem we've got now is that PRP want their papers, which I had already agreed that we would send to them. And the bigger issue is not, we haven't quite worked that through yet. And that will be on issues that are not so relevant to the PRP journal. They're more interested in the biology, the pharmacology, and maybe a bit of pharmaceutical policy or drug policy, but probably not on some of the aspects we're looking at. So I talked to Gary to see whether, and it was getting quite confusing, I think, as well, so many of us, and quite different, even though it's really good coming together and chatting and learning from each other. But in terms of trying to bring a project to an end, probably it's not an effective working relationship. And we wondered if we should either have two meetings. So one is the group writing for PRMP, and the other is the group doing a much more systems approach review, and that could actually quote the work in PRMP if that was helpful. But it would mean that we actually have, it's much easier, I think, to have like a structure of completion and publication and dealing with the translation if we split it up. And I did write to the people that I thought, whose papers I thought would go into the PRMP the other day. I think there's about 4 papers. And so really what Gary and I think we're quite keen to do is just to bring that idea back to everyone, because obviously it's a team approach, and just check that everybody else is in agreement that we split it up, and that enables the group to work more efficiently on meeting the deadline. And the other reason for that is that the COVID moves on and people are busy and they get other roles, and this group can't really meet in perpetuity, though it might be nice. We probably do have to come together, achieve something, and then maybe come back together at another time. So it's kind of a way of wrapping up the project, I guess, as well. We didn't talk about that, Gary, but it was actually my thoughts after we finished. So sorry for adding that in. Anyway, maybe you want to add into that, yeah, Gary.
Yeah, no, that's all good, Jenny. No, appreciate that. Yeah. So what I'd like to do is I'd like to share with you the thoughts on how we structure the papers. So
Yeah.
It's the same set of papers, but the idea is that we publish them in separate journals, but referencing each other. So, you know, I went through this last time. So this is how the publications cover the full power spectrum, essentially. So the paper one, which is essentially the motivation and the explanation of the big picture, the intent is to have actually 2 papers which are essentially covering the same kind of purpose, but interconnected. So the overall situation, themes, and conclusions, with more of a biological emphasis, we're going to PRP, and then the overall situation, themes, and conclusions to go into the SRBS, which is we think the best journal to go for, which would have more emphasis on the social and systems perspective, and this paper would reference this paper, because this one in the timeline would actually come first. So, Jenny, could you just give an indication of when you're looking for let's say mature drafts of these papers?
Yeah, well, for the PRMP one, I think we had suggested end of June, which is actually not that far away now. So that may be an issue. We may have to push that back for a bit, because it's better to get the work done.
I can't possibly do it, Jenny, by the end of June. Just couldn't. It's a dog's breakfast at the moment.
I mean, that's all good. You just have to give me what your deadline is, and then once I know what people are thinking, I can make a decision on that. Yeah.
I mean, I don't know where the others are with the PRP papers. Maybe.
Yeah, we'll go through progress, and in terms of where we think you are and timing on the papers, on a second pass. Just let me go through kind of like the overall direction first. So this one, which would be the COVID narrative, past, present, and future, that one we would target for the SRBS. So this is the paper that Felix is working on with Janet, and a little bit of help from myself. This using system science to enable better solutions paper, that again would go into the SRBS, and this would also take advantage of a paper that I'm working on at the moment, which is going into the Journal of Systems Thinking. So there should be a lot of reuse and reapplication for that. This paper, which is about this is the mammoth paper, the host response and potentials for intervention, that would go into the PRP, and we might potentially have to look at splitting that up. Yeah, but let's talk about that when we get to more detailed discussion. The long COVID paper, that would be going into the PRP as well, and perhaps there might be some opportunity to move some of this content into this content.
Well, I don't think you can understand long COVID till you understand paper, the other paper.
Yes, yeah. There's the viral perspective paper, which again is PRP. There's technologies enabled for healthcare that could go into the SRBS, and again this would make reference to the medical pharmacology from the other papers, because there is still an overarching theme and interconnection between all of these. It's just a case of we need to focus on the more technical medical part first, in order to meet the needs of the journal, but also having that technical baseline for us to work on and make the connections to these more social orientated papers also makes sense. So there's then the realizing collective societal wisdom, which is with Bruce and I think Janet and Bill and also others will be supporting that. And then there's the balancing perspectives, which would essentially bring all of these things together in terms of an integrated summary. So that's the overall idea in terms of how we split up the papers. The more pharmacology, biology ones going into the PRP, and the more social systemy ones going into SRBS, and also reflecting on the prior work that has been done in the PRP. I hope that makes sense. Now the question is, where do we go from here in terms of progress? So
Can I raise a
go
problem?
ahead, Eugenie. Yeah.
Nowhere in the PRP papers are we addressing the interaction of the biology of the pathogenesis or pathophysiology of COVID with repurposing drugs and drug targets, potential drug targets. I don't know whether you're going to do that with long COVID, Bevan, but it would seem to me that that was a critical interface from a therapeutic point of view.
Yeah.
I'm dealing more at the moment with the fundamental biology, hopefully to point out pointers that which you could attack with a drug.
Yes.
I will not pretend to have the expertise to tell you which drug.
Yeah, for me
Eugenie,
that would make sense. That would make sense to have the drug potentials with Bevan's paper.
I might pick up on that. If you look at how the virus has gone through its tropic responses, in other words, how it's achieved its maximum ability to be infective, there's no question in my mind it's done that by mutating in a way that it can get to the upper respiratory tract. That's where it becomes most infectious. And so the issue of repurposing, particularly around RAS and other areas, is, to my mind, very much dictated by the stages of the disease. The first principal stage really being upper respiratory. And so there's a set of repurposed drugs that are suitable for that and are now coming out. Actually, some of the studies out of Oxford are going down that way. Then there's a set of drugs that handle the next stage, which is the lower respiratory tract pathology that flows from that, and those drugs happen to be heavy duty drugs like dexamethasone and so on and so forth. And so I think we can pick that up as we go through with most of the papers. So
yeah.
But you don't think it's necessary to have a poor man's guide to what drug will work where?
I think yes, I think that's true. I think it's not only where, but it's also the when. So it's the where and the when.
Yeah, there is a when, I agree completely.
Yeah.
I don't
The-
think in a way we should be talking renin-angiotensin system, by the way.
Yeah.
I think we have to bring our arguments down to angiotensin and the AT1 receptor versus
Yeah.
ACE2.
Yep.
Renin
Happy with
is
that. Yeah.
renin is a bit irrelevant because you've got
Yeah.
chimates, cathepsins, God knows what else.
Yeah.
Yeah,
No,
if I
I
could,
don't have a
if I
problem
could
with
just
that
share
at
the
all.
screen again briefly. Yeah, so this paper here, which is the implications of long COVID, you can see how I slotted that onto this particular area in the matrix. And what we're really looking at here is the potential utilization and of resources and processes to address this essentially COVID problem, you know, from a practical perspective. Yeah, so the emphasis was long COVID, but this is really all about, you know, agreements on the structures and processes necessary to treat the disease through its full life cycle. Long COVID being, you know, very important, but actually, you know, the earlier stages as well. So, so I see that this is going to gather information from Eugenie's paper here, but also
Yeah.
from this human and virus interaction paper here. So that's how I see things fitting.
If I could maybe just comment on that, I think what is often useful is the counterfactual. In other words, to say in 2020, if something different happened that ended up in us not really meeting, that there'd be no purpose in us meeting because it all worked. What would the all worked be? Now, the all worked tracking backwards is the vaccine, or vaccines rather, coming through. But there's a big chunk there, and that goes to Eugenie's point, and that's the repurposing. In other words, the bit that was missing was the treating the host with existing repurposed drugs and having mechanisms to rapidly define what they would be was the bit that was missing in 2020. If that was there in 2020, in other words, time was bought so that the 20% of people who become ill could now behave in many ways like the 80% who don't, then that would have bought time for the development of the vaccine and presumably antivirals further down the track. And so that to me is the counterfactual, and it goes to what Eugenie was saying, that the big bit that was missing was the repurposing.
Yeah. And I think that relates back to essentially a lack of established wisdom appropriate for understanding these kind of complex, time-orientated situations. You know, apart from a few individuals, very few people were taking that perspective.
Yeah.
I think, to be honest, Gary, it comes back to the concept of everything being in silos.
Yes.
And integrating into systems. So we go backwards and forwards along a very similar track all the time. It's just all we need to do is flesh out how we're going to get the best bang for our buck.
Yeah. This is well illustrated in the study that's come out of Oxford on budesonide, where they started by looking at the observational data, in other words, the administrative data, and found those individuals who were taking that particular drug as an aerosol did well in terms of those people that didn't. Based on that data set and set of information, they then were able to design their randomized control trials in a very, very sensible way, because it was already building on the existing data. So part of the siloing has meant that we haven't been able to access information, critical information, that would help one design very, very sharp experiments for repurposing.
Yeah.
Sorry, I think there's also another aspect to the big picture. And I have to say that I'm a sociologist, an anthropologist. I know nothing about the inner workings of how a virus impacts on the human body, other than what I read in the general media. And certainly, I think it is right. One of the speakers was talking about the importance of circulation, and I believe in South Africa, one of the medical doctors talked about the importance of drugs to ensure good circulation, and it worked. Why it worked, no one knew, but it worked. So it was just practical. But getting to the part of the big picture that interests me is that epidemics in the past have very often been linked with human-animal interactions, and whether we're talking AIDS, swine flu, bird flu, Creutzfeldt-Jakob's, mad cow disease, those kinds of interrelationships and relationality across different parts of living systems needs to be thought through. And of course, the question that is going around is, why did COVID come about? Is it because of the impact of destroying habitat, as Jane Goodall and Attenborough suggested? Is it because of confinement and bad farming methods, which has been suggested by Donaldson and Kimlicka, work on the need for good habitats for animals, whether they're domestic, liminal, those that share our city spaces, whether we're talking agricultural animals or wild animals. And they talk about the importance of protecting habitat for all. Or it's been headline news in Australia because Scott Morrison asks the question, how did it come about? And the obvious issue that has emerged is that in actual fact, there was joint research. And if COVID came about, it was the result of collaborative research actually across nation states. So the change of function aspects and laboratory management probably needs to come into the picture. I believe France was very fed up with the US, saying that they believed that some of their laboratories were better than some of the laboratories that were chosen in China. But in actual fact, the bottom line is that the issue is an international issue, and even if change of function research was responsible, nobody's sure, we were jointly responsible. We being the players on the table who've been pointing fingers vociferously at each other. In actual fact, there is co-determination. So then also part of the big picture needs to be the philosophical departure of how do we actually approach looking at pandemics. We need to be objective. We need to look at perceptions, subjective perceptions, some of the ones I've been sharing. And we need to be intersubjective. We have to come up with a dialogue around how can we develop shared meanings to create new architectures, most basic, having good architectures to protect laboratories and to make good decisions as to how to do research, that kind of stuff, but also good architectures for managing and protecting populations. And apparently there's a lot of research that has come out to show that sections of government that really should be dealing with that sort of thing were dismantled, and it's applied across the globe. Very poor management. So really the COVID pandemic focuses the mind on social, cultural, political, economic, environmental aspects, and the system sciences are beautifully placed, or our organization, to perhaps have papers where we're all experts in different areas. And Gary's done this brilliant sort of overarching schema. And maybe we should actually write to our our expertise and cross-reference our papers where we can. I think that would be best, because how can I explain aspects of philosophy to people who are experts in biology and aspects of medicine, which I know nothing about, but I can get a sense of the big picture by navigating across the different experts talking in their field. So I think it would be brilliant if a journal could follow, or journals, two special issues could do what Gary is suggesting. And perhaps the way forward is to allow us to do our own thing and contribute to your shared vision.
Yes, that's brilliant. Thank you, Janet. Yeah, that was beautiful, because the way you described it, you started off with the biological and the pharmacological, and then you gradually moved into the political, social, cultural aspects. So that's exactly the kind of blend we're looking for. So that first part of that you were talking about, I think, needs to go into our paper 1 in the PRP, and then the second part needs to go into the papers that we're going to go into the SRBS. So and what you were talking about in terms of the collaboration across the two areas was perfect. So thank you very much for that. That was great. I think I'm anticipating what Richard is going to be. I've not seen your slides, Richard, but I'm imagining that your slides are actually going to touch on both of those aspects. The technical pharmacology aspects, but it's also going to talk about, I would imagine, the bats and the movement of the virus from the bats into the human situation.
Yep.
So
Yep.
would you be willing to go through those slides and give us the story? Because I think everyone will be interested in that, and that helps to bring us all together.
Yeah. If you can put it up at your end, that would be terrific.
Oh, okay.
Yeah.
Hang on a second. Hang on a second.
So while you're doing that, I might just pick up on
Please,
the anthropological issues that Janet was just referring to, because they're fundamentally important. And if you look at humans and viruses from that perspective, you see that humans went down one path and viruses and pathogens went down another. And it's fairly easy to see the paths they went down. Humans went down a path of multicellular organization, where viruses, and to a certain extent bacteria, went down a path of single cell or virtually minimalist approaches to replication and reproduction and so on and so forth. And what we're looking at really is the intersection between those two things. And we're looking at the intersection of something that was really rolled along millions of years ago. And so, and you're perfectly correct. We have no mechanism of bringing all that together to understand how those points of intersection occurred along the way and where they are now. But I'd like to start the issue that's fascinated me all the way through is, what is the fundamental driver from cell to cell to cell, or from human to human with this virus? This is not like a bacteria, as I said before. It's not like other pathogens. It's not carrying its own oxygen supply. It's not carrying its own ability to produce its own nutrients and so on and so forth. It's very parasitic on, in this case, humans, but mammals in general. But because because it doesn't have those characteristics of life and has more characteristics of chemistry, if that's the case, it must be obeying the fundamental laws of chemistry. And the fundamental laws of chemistry are the laws of thermodynamics that were put in place 100 years ago, undefined about 100 years ago. And so I'll just run you through a little bit of that, and then we'll come out the other end in terms of the biology. So the next slide, Gary. To me, if you stand back and look at, if you were a Martian and came down on Earth and looked at the moment and said, well, what's going on? Well, there's a lot of issues around the biology, and there's a lot of issues around pharmacology and societal issues and so on and so forth. But if you distill it to its essence, it comes down to the vulnerability of human complexity versus the lethality of viral simplicity. There's nothing simpler in many ways than a virus. It's got 4 proteins and a bit of RNA and a little bit of lipid. The vulnerability of humans is because we're multicellular creatures that we've been refining the ability to regulate multicellular processes over thousands and millions of years. And the price we pay for that is a vulnerability, and let me come to that right now. Each of us has about 42 trillion cells. And they're all organized. None of us is sitting around this table at the moment saying, I'm monitoring my cardiac output, I'm monitoring my flow through the kidneys, I'm looking at glucose handling in my liver and the rest of it, because we've evolved processes to do that, regulatory processes to do that. And that's part of our complexity. We've had to do that to fundamentally free up our CNS. In terms of the virus, as I said before, it's incredibly simple. If you said to yourself, what does this virus have that makes it so lethal? If you distill it to its essence, it's a surface. It's almost architectural. And in other words, it just has a surface, but it has the ability to change that surface at great speed. And in a chemical sense, the ability to change that surface so that it'll drive other reactions. And those other reactions are in the human, and those other reactions are around the human vulnerability. And so you can't get anything simpler than a small surface. Now, if you looked at that surface, you said, well, there's 4 proteins only in that virus. That virus is 1,000th the size of a single human cell. And the protein that we're looking at there is barely 1,200 amino acids long. It's not big. And the binding area is about 100 amino acids only. And within that, there are probably only about 6 or 7 amino acids that are important. And so that you've got this incredibly tiny surface that's the power of the virus. And so what I've been trying to understand more clearly is, as the virus moves through the human, how is that issue of surface interaction driving that process? There's no energy with this virus. There's no equivalent of mitochondria. There's no equivalent of running free radicals off ferric ion like you do with bacteria. If it is chemistry, then it's got to adhere to the fundamental laws of chemistry. And the fundamental law of chemistry, one of the key ones is on the next slide. And I apologize for the equation, but most of you, or many of you probably remember it from school days, and it's a thing called Gibbs free energy. Gibbs free energy drives every cell in your body and drives your whole entity. And it's effectively Gibbs free energy is the energy that is associated with, not in our case, living cells, but it's also associated with the interaction between chemistry and our living cells. And there's a thing called the Gibbs free energy, in other words, that delta G there, which is dictated by 2 things, enthalpy and entropy. And if I can just describe those 2 things, because they are fundamental in the universe, fundamental on this planet. The enthalpy and entropy, very hard to describe, but if you've got teenage kids, it's quite easy. If you have a teenage kid who has a neat room, then you can say the entropy levels are low. If you've got a teenage kid that has a normal room, which is in absolute chaos, then the entropy levels are high. And that's, in other words, the degrees of freedom and everything else are through the roof. And so it's those 2 things that actually dictate Gibbs free energy. Now, a spontaneous reaction, which could be a virus binding to one of our cells, occurs when that Gibbs free energy is negative, never positive. It has to be negative. And it says then that reaction can occur. So you ask yourself, okay, if this virus binds to a cell, is the Gibbs free energy going in the right direction? Is, in the lack of anything else that's out there to drive that process, is this working? And again, next slide, please, Gary. Now, if you look at the right-hand side of those two diagrams there, to make a product, and the product could be the binding of the virus to a angiotensin II, for example, receptor, which is how we get infected, then what Gibbs free energy says is for that to occur, we have to have reactants and products. The reactants is the virus interacting with the receptor on the angiotensin II, and then ultimately leading to products. In this particular case, it will be the binding to the receptor, replication of the virus within it. But the critical thing on that diagram is that red line where delta G is less than 0. Because if it's less than 0, it means it's a spontaneous reaction. It will occur on its own. It doesn't need any prompting. It doesn't need any help. It'll occur on its own. Next slide. So if you look at the, if you look now at what we're dealing with all of our cells, our 42 trillion cells, particularly those that have got ACE2 receptors, you've got a virus that I've shown there binding to that blue thing, which is the receptor, the angiotensin II receptor that Eugenie's been talking about. You then have entry into that cell, and then you have replication of the virus within that cell and lysis and the way it goes. So if you looked at the chemistry of that and said, this is not a living system, this is a chemistry game here, then you see two things. There's an equilibrium thermodynamics in terms of binding. That virus can bind on and can bind off the receptor, depending on the numbers there and the circumstances and so on and so forth around it. There's also a non-equilibrium thermodynamics where you have replication and lysis. In other words, once the virus goes into the cell, based on entropy alone, it can't go backwards. Once it replicates, it can't unreplicate itself and then go back. It can only go forward as entropy expands anyway, and that's part of the issue. So the question then becomes, are both of these processes, which are thermodynamic processes, dictated by changes in Gibbs free energy? And if they are, they both must be negative, if that's the process. Next slide, Gary. Now, we're not clearly the only ones thinking about that line. Popovic and Miseva have taken this thinking to SARS, MERS, and SARS-CoV-2. SARS being a very similar virus to SARS-CoV-2 that came out in 2002, MERS 2012, all coronaviruses, the three, but very different in their potency, but nonetheless coronaviruses. What they did is they said the following: that viral multiplication at its core is a chemical process characterized by a growth reaction, and the Gibbs energy, a predictor of a spontaneous reaction of growth through non-equilibrium thermodynamics, enables the growth comparison between the host and the virus. In other words, if the Gibbs free energy is negative with respect to the virus and the host, then that reaction is spontaneous. It needs nothing else, but it will just go on its own. So they did a comparison of SARS, MERS, and SARS-CoV-2 in terms of measuring or looking at the Gibbs free energy and the host cells. And they came to the conclusion that the Gibbs energy of growth of the virus nucleocapsid was more negative than it is in the host. They then suggested that there's a greater spontaneous virus multiplication rate producing an enhanced reservoir of virus, permitting a greater population of transmission. And so they were saying that you could now look at the whole concept of the virus replicating as straight chemistry dictated by the laws of thermodynamics that cover every aspect of chemistry in the universe, fundamentally. Next slide, please. So if we now say the replication and lysis component alone is running by Gibbs free energy, then the other question we have to ask is, well, this wouldn't occur unless the binding is also running in the right direction in terms of Gibbs free energy. And because you need the binding to ultimately have entry into the cell and then replication and lysis. So next slide. So we're not alone again. here's Coley and Chowdhury, and this year, in fact, a couple of months ago, they've actually now measured the whole issue of Gibbs free energy. What they've done is constructed all of the ACE2 binding sites in a whole series of species of animals. And by constructed, they've done that in silico, because you can do that now. And the delta G there is the Gibbs free energy, as you can see, and right at the top of the pile is the human. And so with all of the species they've looked at, and there's more than those that they looked at on this slide, you can see number one, they're negative. So it's fitting in perfectly with the Gibbs free reaction, saying this is a spontaneous event. In other words, it needs nothing else, but it'll be driven by entropy and the balance of entropy and enthalpy. And by the way, when you look at that, humans, by the nature of the tropism, by the nature of the structure of the ACE2 target, has got a Gibbs energy that is very negative compared to other species. So the next slide, Gary, if you can go to the next slide, which shows you can actually now look, if you look at the extreme left-hand side, the delta G bind, that's the Gibbs free energy again. Look at the virus that was out there in 2002. You'll see that it's about 12 kilocalories, and the SARS-CoV-2, the current one, is about 31. Now, we know that SARS-CoV-1 is not as potent or as damaging or as infective as the current virus. And there you see the current virus is more negative in terms of Gibbs, in terms of Gibbs free energy. So that fits. So then the next slide, Gary. It's the final slide. And so what this is telling us, if you remember some months ago, I was starting on KD, which is a major part of this, but this is now pushing it further down that chemistry road. But the spontaneous reaction occurs in binding and replication because the change in Gibbs free energy is negative. It's negative in both the binding and in the replication. It will occur on its own. It doesn't need anything else except a high affinity site to bind. As soon as it binds, the fundamental laws of chemistry say this will go, this will happen. And so that's, Gary, that brings in where it is. So we have one of the simplest things on Earth, 1,000th the size of a human cell, tackling something that's got 42 trillion cells in each of us. And it's been driven by the fundamental laws of thermodynamics, which, by the way, were put in place by some Englishmen, Boyle and Co. in the 1800s, in terms of defining what those fundamental laws are. But they've defined everything in terms of chemistry. So I think, Gary, what it's telling us is, as we map the passage of this virus through the human, through the upper respiratory tract, going to the lower respiratory tract, we've got such a foundation to say now that this virus has the ability to spread fundamentally by spontaneous movement based on the laws of the fundamental laws of chemistry.
Can we ask questions, or maybe put some thoughts about this? So two questions I have. Well, two thoughts. So number one, can we control the Gibbs free energy? Can we make it positive?
Yeah, it's a great question. And I'm convinced those that are looking for drugs that would, or entities that would prevent the binding of SARS-CoV-2 to the receptor, will be looking for those drugs that impair that Gibbs energy. In other words, if you can define your drugs such that you'll make that Gibbs free energy more positive, then you'll have something that should be very useful in preventing the drug and preventing the virus binding to its target.
And the other thought that I have is that, because my feelings is that the main purpose of this cohort and the papers are doing is to show how system science can help in situations. So taking your situation, for example, that doesn't mean like, shouldn't we look at concepts from systems and see how these concepts from systems help us to understand? So one of the concepts that could help us to understand this Gibbs free energy is maybe the term synergy. So if a virus is in synergy with its environment, then you'll have this potency. If the virus is not in synergy with the environment, then it's not potent. So do you think it's
let's
relevant
pick that
for
up.
us?
That's fundamental. The other thing that's happened from an anthropological almost standpoint is as humans have gone down a multicellular road of where they have focused on one thing, and that is reducing the effect of mutations in our own replication machinery. The virus have gone down a simpler road where they say, we're going to have no repair mechanisms whatsoever associated with our nuclear RNA in this particular case, or non-nuclear RNA, just RNA, and we're going to embrace every mutation possible. And so in terms of what you're saying, what you've got fundamentally is a piece of chemistry that rapidly changes that surface composition through mutations. And all it has to do is change enough times until it gets a boost along with Gibbs free energy. That boost along is what we're calling variance. And in other words, if you get the right sequence, you'll drive the Gibbs free energy more negative, and then you can guarantee that a spontaneous event will now occur in terms of binding and entry and replication. So.
The interesting part that I see here, how this knowledge is transferred.
Wow, it's quite incredible. I've become I've come to appreciate that our human-centric thinking here is tripping us up. In other words, we associate intelligence with neurons. I can associate intelligence quite often with non-neuronal entities, and this is one of them. This is, you stand back and look at this and say, well, it's not really intelligence as we know it, but it's achieving a chemical output, and that chemical output, frankly, is the fundamental issue of Gibbs free energy. Gibbs free energy is not something just around viruses, drives everything. You talk to people in astronomy, they'll talk about the universe in terms of Gibbs free energy. So it's one of those fundamentals.
Thanks. Eugenie, you had your hand up.
Yeah, I did. First of all, just because I'm very naive, surely the binding of any molecule to its receptor is a state of that process of negative free energy for it to occur. It's the same chemical process. Am I right?
Yeah,
and
you're correct.
Secondly, one of the worries I have is that for the virus to replicate involves an active process by the enzymes TMPRSS2 and ADAM17. So how do they do everything thermodynamically the right way around?
It's a terrific question. I haven't seen anyone look at Gibbs free energy of that intermediate stage between binding and replication. In other words, the entry that you're talking about. But the entry that you talk about can occur in two ways. It can occur through the protease that you just described, in other words, the cleaving of the S protein, but it also can occur just quite efficiently through well, if you have the cleavage, you usually have the fusion with the membrane, and then the whole lot's dragged into the cell. But you can have endocytosis working as well, regardless of that. So you can have multiple ways in. But part of the reason for my thinking here is the following, Eugenie, and it comes from years and years of seeing things but not really seeing them. And that is, I used to watch all the time, and I did it myself, people with organ bath experience where they'd add dose after dose after dose and get a dose response curve and a response. But let's take a simple example. Let's say it's adrenaline on a myocyte contracting in an organ bath. So the adrenaline goes around to the beta receptors of the heart, you now decrease the Gibbs free energy on that and so on and so forth. So you've got the binding. But then you've got this transduction process that goes on. It could be cyclic AMP. Then you've got contraction of cardiac muscle, for example, and the rest of it. But what's truly astonishing is all of those processes subsequent to the binding are slaves to the initial binding process. In other words, whatever happens up front is what you're going to get at the end.
That
And
is so cool. Yeah.
And I see that in the cancer field. Jenny, I think, is here somewhere. And this was first described in the cancer field. In other words, so when I dug it back and looked at it a month ago, with tamoxifen, where people made the observation that when they added tamoxifen and it bound to the estrogen receptor, the cells faithfully followed to the concentration, the apoptotic response or whatever it was that was going on. And so there's a rule that there's something that's there that we haven't described well in biology that says that initial binding is king or queen. It's dominant in the process. And I think part of the reason it's dominant is that really if you dig into the Gibbs equation, it's a balance between entropy and enthalpy. And there are 4 options you've got in the universe for those balance of those 4. I think it's such that the binding is a set of options that is absolutely dominant in the whole process. Anything that occurs beyond that is a slave to it.
There's just one other comment I'd make, is that it has chosen as a receptor that's absolutely ubiquitous.
Yep.
And is quite frankly the driver of the appalling host response that occurs with infection. Take that receptor. If that receptor is so critically important to the control of inflammation.
Yeah.
I would argue, therefore, that you said SARS itself wasn't particularly lethal. It wasn't infectious, but it's lethal. It's lethal. Yeah,
absolutely. Now, and so let's tease that out because it's fundamental. What the Gibbs free energy would say is that the more negative you are, the more you'll propagate that chemistry, that reaction, that curve. The easiest way to stop that curve is to kill the host, and so the most effective virus in many cases will be the one that can mutate to maximize infectivity, but not necessarily to have high mortality.
Yeah.
And that's what this virus has done. And in other words, this virus, by mutating to a point where it can bind in the upper respiratory tract, so we all sneeze on everyone else and we spread it like fury. Even worse, half the viral load is in the upper respiratory tract before we're even aware of it, and let alone detect it. So that it almost says that what happens in the lower respiratory tract is pathology that's indifferent to what the virus wants.
Yes.
You know, that's just
a nice way of putting it.
Yeah.
But very, very deleterious to the host.
Oh, unbelievably. But in terms of a population, where the systems analysis now kicks in is because that sits in the 20% of individuals who become ill, in other words, where you go low respiratory tract, but because it's carrying the high infectivity because it's mutated to the upper respiratory tract, then it spreads throughout the world. And so the total numbers of people that have this terrible pathology are just enormous.
Yeah. So what I'd like to do before we close, in let's say in 20 minutes, what I'd like to do is go through everyone on the call and see where they are on their papers and if they understand essentially where we are in terms of the emphasis. You know, the emphasis at the moment is going to be on the biological medical papers, But that doesn't mean that the other papers aren't important. But there's going to be, I think, there's going to be a lag between the first set and the second set, and that lag is also necessary so that we can take advantage of what has already been written. So let's just go across as I see people on the screen. So Eugenie, do you have more of an understanding now of what you need to do and how you fit?
I'm happy about, I think I will continue down my path. I think what Felix sent me today is a sort of, would almost be a conclusion to, you know, explaining the biology, building my arguments, and then putting it back into what Felix has sent as diagrams, which will take a degree of fiddling, Jenny. And I'm panicking about time because I've still got I've still got some work to do going down the path. I'm up to, you know, I think I've got signaling right. Now I've got to get, ACE2 counteractions to angiotensin 81R, and then I've got to go down and talk about specific biologies because of the diversity of responses when you and the ACE2 knockout. And hopefully then I want to go back and pinpoint points at where you guys can talk about drugs.
Yep, I don't think nobody wants anyone cutting corners here, Eugenie. So the thing is probably just for you to let us know what sort of time frame you're aiming for. And the only reason is that in a lovely idealistic world, we wouldn't have any time frames and we could just take as long as we needed, but the journal has got deadlines for stuff. And so we probably just need to be realistic, I think, with how long it's going to take us, and then send that back to the journal. And look, if it's too long for them, that we could just try somewhere else. But I think it's better that we are realistic about how long it's going to take us to do it. So maybe if you just let me know what sort of time frame you're aiming for, that's probably helpful.
Yeah, I've got a lot of time, you know, I've got to put the references into EndNote and all that sort of stuff as I'm going, and I've got to design figures as well. So there's a hell of a lot of fiddle faff.
Yeah. Also, maybe like August is better for you, do you think?
Oh, that'd be a great relief, because I am going to have a holiday as well. I confess.
Yeah. Richard and everyone else, do you think August is a reasonable time frame for me to, because it's best that I just write to them once and say that our revised time is this, and this is what our papers are going to look like now?
From my perspective, that would be yes.
Okay. Cool.
Okay, so while you're speaking, Richard, you know, where are you, do you think, on your paper at the moment in terms of content?
I'd be halfway there now. What's happened is I got to a point where I was asking myself more questions as I was writing, so I had to backtrack, and that's when I ended up with Gibbs and Co. Yeah. But I think that's the nature of the game. And but so I'm trying to go from the notion of how the virus got into a human, at least based on affinities and so on and so forth, where it will be in the human. So at the moment, I'm well and truly now in the human, but Eugenie, I've got a tic tac with you, because I'm not going to go down the level around the fine detail that I know that you're covering around ATR1 and so on and so forth. So
no, well, I mean, I'm assuming that it will be complementary. I have to say, the question of what level of complexity I go to when I get down to different organs is bugging me a bit, because in every organ you've got multitude of effects. Yeah, I mean, just taking the heart with hypertrophy versus fibrosis, etc. Yeah. As for the brain, well.
Okay. Right. Thanks, Bevan.
Right. Well, the hypothesis I'm working on is that after the virus infects the cell and replicates itself and leaves the cell is permanently damaged and has oxidative stress. Yeah. And this could be the basis of the the long COVID, which
Yeah.
happens to people even for mild infection.
Yeah.
And it lasts for ages. And this is not just with COVID-19. It's happened with many other viral infections, like going way back to glandular fever and hepatitis. And so I think it could be a general phenomenon that the surviving cells after the replication of the new virus is damaged and has oxidative stress, which means free radicals are generated, which keep on causing damage. And my thesis is that NRF2 is a very important transcription factor, which if you can activate that, it will restore some of the cytoprotective enzymes that make, say, glutathione, which then quench the free radicals.
Yeah.
And there are drugs that can be repurposed to be NRF2 activators, which may well be worthy to be trialed in the patients with the long COVID. And probably the safest and the simplest one is probably melatonin. And it is certainly known to activate NRF2. And there is a view now that it will overcome some of the symptoms of the long COVID, like sleep disturbances, and it resets the circadian rhythms so that the person has a good sleep, wakes up refreshed, and feels much better. So I'll probably concentrate on just melatonin as a repurposed drug, which is ready to be used because it's quite a safe drug. They've given grams of it to people in the past without any effects, any adverse effects. So I can build a story around that. And there are cases in UK of they've been using the melatonin to nurses and doctors who have had disturbed sleep from the long hours they're working, and it's actually apparently quite good with chronic use. It's not just a once only effect on. It's not a hypnotic drug, but with continual use, you will start to reset your circadian balances, and probably it's all through activating NRF2 and restoring the antioxidant products of the enzymes.
And of course, as you age, you reduce your melatonin production, which is why your hair goes white.
Ah,
Well, we can try that out ourselves.
yeah. Thanks,
But
Bevan.
It's called, they call it circadian here, but it's really circadian.
Yeah. No, the drug is called Circadin. That's its trade name. And that's it's just become an S3 drug, so you can buy it over the counter tomorrow without going to your doctor. It's only $30 for a month supply. but anyway, I think it's something that needs to be brought out to the rest of the readership.
I think that just follows on from what my thinking.
Yeah.
But I think Eugenie and Kirsty sent me a paper from the Sydney group of the Kirby Institute, and they're saying that they've got quite good evidence that the long COVID people have got inflammation, chronic inflammation.
Yeah.
And that's why I think it ties in with activating the NRF2 to generate free radicals, overcome your low scale inflammation.
Yeah.
And
So
angiotensin is a major driver of the production of those reactive oxygen species, and angiotensin is in every single cell you can look at, involved in inflammation.
Certainly, it's pro-inflammatory, the A2, and but I wasn't aiming to discuss that. I was just going to talk about overcoming the oxidative stress with using NRF2.
Well,
but you could come in and talk about A2 as a secondary involver.
No, I'm coming down the pathway of angiotensin 2, reactive oxygen species, inflammation, attracting cells that have angiotensin 2, and the cycle builds up and builds up until you've got a rip-roaring inflammation.
Yeah.
which is no longer controlled by ACE2.
Right.
Yeah. So Janet's had to drop off, but I'm in touch with Janet and Felix on their paper. They're making good progress on that. Bill, do you have any thoughts at the moment? You need to keep it short though, I'm afraid, mate. You're on mute. You're on mute. Off mute. That's
Okay.
it.
I made my career on this discovery that if the World Bank started to look at it as projects instead of looking at it as one circle that had to be brought under control. So here's the project, we have to bring it under control. And if they added a circle of influence beyond that, of things you couldn't control, where you had some influence over, and then on top of that, you added another circle of things that you couldn't control or influence, but they affected you. So, and intellectually and so on, that was it, to produce a revolution in the way projects were designed. What I'm looking at now is to say, yes, there are those, but you have to apply the same thinking to each of those. So now there are 3 by 3, so there are just 9 things. So I'm looking at the world through these 9 things. And what it means in terms of these papers is the step I have to add is the step of taking whatever is in paper 1 and go to the last of them and get them to talk to each other, because they're complements, they're the opposite of each other. You know, one's very open and whole, and the bottom one is very closed and finished. So they're complements. And so if in our design processes, in our thinking processes, we're able to do that for all our 9 variables. So the first step is to get the 1 and 9 to talk to each other, with the 1 in the middle, the 5, doing some mediating. And then you come to 2 and whatever's in 2 and whatever's in 8 to do the same thing. And then whatever in 3 and whatever in 7 to do the same thing. Just as that simple process of 3 was enough to get a revolution, just doing that complementary organization in design. So that's what I'm testing out. And as long as there's material in paper 1 and in paper 9, I can do something, you know.
Right. Thanks, Bill. Bruce?
Good morning, or
Good morning.
good evening, wherever you are. I've done an outline, but I think now it needs to be fit within the rest of the papers. And I'm very interested to talk to Janet about her anthropological view and see if we can kind of merge them together. But I'm very open and happy to shape which in whatever direction is necessary.
Great. And I see a correlation with what you're doing at the moment with your paper, and also what you're doing with the presentation at the ISSS in a few weeks. So lots of reuse and synergy going on at the moment.
Yeah. And by the way, I really appreciate the discussion on biology and chemistry. Yeah. And I'm going to be looking for Gibbs free energy interaction with cultural communication.
Yes.
Thank you.
Yeah. Thanks. Arnond, hi there. Sorry I've left you till last.
No, that's okay. Yeah. So I have an outline. What I started doing is I started looking at it as different class of problems, and more I read the other papers, I feel that all of them are different pathologies that needs to be taken into account. And then we need to then work on what would be the architecture for a digital healthcare system that can help deal with these pathologies. So I mailed a slide deck, which is a refinement of the ideas I had worked over the last couple of weeks.
And how are things in India at the moment, Anand?
It's crazy. We are still, I mean, they say lockdown is lifted, but still nothing is lifted. And the only thing we are doing is we are putting our head down. We are not going out. Just continuing to maintaining the vigil, because it's pretty obvious if you go into an hospital, you are not going to come back. Pretty much clear.
Yeah.
You don't get anything. You don't get anything. There are no resources, and I mean, then people have started making the decision whether government hospitals are crowded. If you go to private hospitals, your money is gone. Whatever you have earned so far, everything is gone. So one of my colleagues, in the last 2-3 months, he lost both his father and mother, and he has lost everything. Whatever he has earned, his father has earned, mother has earned, everything is lost. All the money is gone because the hospital just kept demanding and taking money out of him. There's no solution. So we are only trying to avoid that.
Can I ask a sort of general question which doesn't relate to what we're writing? And because there's Anand and all you guys in the UK, it seems to me that vaccination isn't going to be an answer in terms of, you know, everybody's going on having to have lockdowns because you can't get to 70-80% of the population being vaccinated. And you've had, I know in England, you've had repeated lockdowns. And here we're being told, you know, the doors will open for everybody when you're all vaccinated. And even if we were all vaccinated to 70%, the doors are not going to be open. Is that a true feeling?
Well, currently in the UK, they've delayed the complete opening for another month, I think, but they are planning on essentially going back to normal in a couple of months. I can't remember the particular date. Sorry, Anand, you were going to say something.
Yeah, I mean, if we actually look at what is this vaccination all about, you have a long chain of molecules or some living organism, out of which you identify one part, which is the spike protein, and you massage it, you inject it, hoping that the immune system will be preventing that spike protein from getting replicated. And now if you do a study of whether the immune cells are grown sufficiently, we see that it's not happening. I mean, my wife says that she underwent a test and she was never COVID positive in the last 1.5 years. But now her results are something like she was already exposed to COVID. And she is reacting when her cells are reacting to the entire genome sequence. So, I mean, we are not confident about the vaccination program. And there is a lot of nonsense going around vaccination. See, there is a vaccination process where they say you use some live cells, something is being used to trigger to start the culture of the spike protein or the virus cells, whatever you're going to put in the vaccine. So now they are saying it is made from calf, made from a cow. So it is holy, you shouldn't take it. So all this nonsense is happening. So, it is pretty difficult. For example, I have a servant maid. She is eligible for vaccination for the last 2 months, and she has not gone there at all. Doesn't want to go. People are very hesitant to go and get vaccinated. So we will just continue operating like this. We have to only look for ourselves. We don't have a choice.
These are all the social
Yeah.
implications. And we also have politicized some of the decisions that people are making. And that's worrying. But I think it's all around the people and their mental models of what they think is actually happening and what is beneficial for them. And there's a lot of that, even health and food and all of that, I would imagine gets factored into this.
Yeah. Jenny, are you okay to sort of sum up? I mean, go ahead.
I wasn't sure there was much more to sum up because I think that the last three probably fit more into the systems review. I guess the only issue I've really got is by the dichotomy of what we've just sort of agreed to do, we're actually guilty of the same problem, which is dividing up the science from all the system issues. So I think that our papers for PRP would heavily need to reference yours and the system ones, Gary. Having just looked at that, the proposed content in your PowerPoint, it looks as if you actually got some headings in some of the PRP papers that mean we could include some of that stuff as well. So maybe it's going to all be okay. So I guess the next thing would be to, it sounds like the PRP people are happy that I asked for an extension till August, and we're pretty happy that we know what we've all got to do. Is that right? You, Jenny, and Richard and Bevan, you were all good? And none of us need to be micromanaged, but we probably do need to just check in with each other to make sure that we're staying true to what we thought we were covering, and make sure that the synergies and so the synergies but not overlap, but also leaving it open to a systems approach so that your papers can reference off ours and tie in the future. Does that sort of sound reasonable?
Yeah. I mean, what I'm happy to do is to keep running these, every couple of weeks.
Okay.
I think obviously the focus needs to be on the more pharmacology PRP aspects, at least for the next couple of months. But I don't think it hurts to have, if the other people are okay, to attend and take more of a backseat observing perspective. I think we can chip in occasionally with insights from a systems perspective, which might be additive and complementary to what you're doing. So if people are okay to work like that, then I think it would work well.
I think there is a converse, Gary. I learn a lot hearing you systems people talk, and that sort of changes how you think about things and makes you less.
I think there's a big systems thing to take into account, is that even in the best of conditions, if you produce 9 papers that do exactly ideally what you would want them to do, you've still got only what I would call an appreciation of the issues. And to move and that's really, you've done a great job of sorting out the information of things. But now to get that real in the social and political world, you need something else, which doesn't come about through papers. It comes about through the way you engage, who you engage with, why you engage, and so on. So I think, and probably the most that this group of people and the papers could contribute to is setting up the first stage of that, is to say, okay, let's take our results and now let's go to some form of engagement. So who are the best people who, if they heard this stuff, have some influence in moving it, not necessarily to solve it, but to move it somewhere, like, invite, my idea from World Bank days is you invite 3 regions, 3 very different regions who might be interested in, and be willing. So your job is to persuade them, but it's not done through the papers get us there, or the mental image of what needs to be done. But then you need an energetic mechanism, which is some kind of conference or some kind of convention that invites not just academics and so on who can sort the problems out as we do, but who have things like money and things, you know, and have connections and so on, who can move it. And eventually those will get it to move even further down to the, and I think of it as 3 levels, you know, the global is the global thinking, and then the energetic is regional political action, and then local is where the actual changes happen. And so it's more or less a 3-step process using different social mechanisms and political mechanisms to move from global thinking to local, sticking needles in or making new drugs or whatever. You know.
Yeah, well, that's funny, because that's in keeping with one of the discussions we had, you know, even before Christmas, when we first started getting together. You know, we said that, you know, doing papers just wasn't enough, and Jennifer and Richard and Eugenie, you know, all of those efforts we'd made in terms of going to the media and stuff, which didn't get us very far.
I mean, I guess somebody like PricewaterhouseCoopers or KPMG or some, if they could see an advantage in what we're saying in terms of their ability to act as consultants or whatever, might be a pathway where we got money.
Yeah, that's so funny, because that's, you know, I've been talking with a friend and say, you know, I'm reaching the limits of what I can do with my career now, and I have to kind of wrap it up and hand it on to someone. And the thing we came up with was, yeah, you get some kind of licensing agreements and so on with people like KPMG or Pricewaterhouse and so on, and that's where you get your money. And of course, they all now KPMG is recruiting 100,000 consultants to consult to the government on exactly the kind of things that would be the background to COVID. You know, how can we get agencies to talk to each other and get a better public health, and, you know, all of those other background things. And now the big accounting firms who know where the money is, they're thinking that that's a prime area for action.
Yeah, so I'm going to bring us to a close in a little while, but Anand has asked if he can just show a couple of slides. So go for it, Anand.
I just present a couple of slides.
Oh.
So what you see here is our old reality. In the sense, just before COVID, look at private hospitals. Usually only people who can spend that much money can go to private hospitals, so generally empty. The government hospitals, the norm was around, in 2 hours, around 400 to 600 people will be standing there for meeting one doctor. And it's not only about hospital, but the situation is there everywhere. Transportation, in a coach where we can have only 80 people, we have some 300 people. And water supply, I mean, just outside my society, this keeps happening. If summer comes in and we see it year after year after year. So again, just outside my society, I can see the housing units, slum areas, which are not regulated. Most of them are illegal, but nobody takes it. And crowded school autos is a normal thing for us. Our children go in such vehicles. It's essentially a 3-seater. And you have some 30, 40 kids inside on their bags. So now this is you can say, a breeding ground for things like COVID, because in this situation where everything is so crowded and the resources are so poor, things spread very fast. So what we see, or what is the official number that's been given, and the actual count, there's a big difference. You can say 10 times, 15 times is a normal difference for us. It's not because they want to hide, it's because we don't have data. So I just wanted to show these two slides to show what is the extent of the problem here.
Yeah, thanks, Anand. Appreciate that.
Horrific.
Yeah, so let's draw that to a close. Thanks a lot, everybody, for contributing, and the discussion is really appreciated. You know, as you can see, you know, this is, you know, really important. But I'll see you all again in a couple of weeks, and thanks, and just continue to correspond by email as needed, and if you need any help, just reach out. Okay, thanks a lot, everyone.
Bye.
Thanks, everyone.
Good,
Bye.
thank you.
Take
Bye.
care.
Thank you.
Thanks, Gary. See ya.